Hello everyone, welcome to my talk. I'm Qiuyu Lian from Tsinghua University. Today I'm going to introduce my work, which is Artificial Cell Type Aware Cell Type Classification in CITE-seq. CITE-seq is a new technology. It concurrently measures mRNA and cell surface markers at single-cell level at the same time. Cell surface markers are proteins expressed on the surface of cells. They are often used as markers in cell type identification. For a long time, they are treated as a golden standard. With surface markers, a biologist can identify cell types following a procedure called gating. Measuring surface markers is very important because we often observe discrepancies between mRNA profiles and surface marker profiles. Below, I'm showing you a CITE-seq dataset of PBMC. In the left t-SNE plot, we have the manual gating labels based on surface markers. In the right t-SNE plot, we have the clustering labels based on mRNA. By comparing the two, we could observe that there are major disagreements. As CITE-seq facilitates accurate cell type annotation with surface markers, our goal of CITE-sort is to develop a clustering method for CITE-seq surface marker data. And we want our model to be easy to interpret, and we want it to facilitate easy annotation of cell types. The problem is that canonical clustering method performed badly in CITE-seq dataset. In the CITE-seq dataset of PBMC, the manual gating labels are displayed with different colors in the CD3 versus CD19 scatter plot, and the label is used as a ground truth. Comparing with GMM and k-means clustering result, we found that none of them could produce clustering results similar with the ground truth. Particularly, none of them could separate this tiny cluster with other clusters. Why do these canonical methods fail? That's because in CITE-seq surface marker dataset, there are special characteristics. The first characteristic is the cluster imbalance. In the CD3 versus CD19 scatter plot, the two clusters in the blue box and the red box differ significantly in cluster size. Secondly, cluster number is huge. If we zoom in this t-SNE plot, we'll observe that there are many tiny clusters. Thirdly, clusters may not be well separated. If we compare the top right cluster against the top left cluster and the bottom right cluster, we observe that there are no clearly defined boundaries. All these characteristics are actually induced by technical artifacts, multiplex. In droplet-based single-cell sequencing, ideally, each droplet just captures one cell, which forms singlets. Occasionally, multiple cells could be encapsulated into one droplet, which forms multiplex. Multiplex could induce artificial cell types. Still in this CD3 versus CD19 scatter plot, we notice that there is a cluster highly expressed both CD3 and CD19. Well, according to the existing knowledge of cell types and their characteristics in PBMCs, we cannot match this cluster with any cell type in the list. As a reference, I'm also showing you a PBMC dataset measured by flow cytometry. By comparing the two figures, we'll find that they are similar with each other mostly, except that there is no cluster which is CD3 positive and CD19 positive in flow cytometry dataset. Then what is this cluster in CITE-seq dataset? In reality, for CITE-seq technology, during the droplet formation process, we could have multiplex containing both T cells and B cells. And when this happens, it will manifest in the scatter plot as a particular cell type. Well, this cell type does not really exist. They are artificial cell types, ACTs. ACTs will impede clustering. Artificial cell types will cause three major challenges to clustering methods. Firstly, ACTs will induce cluster imbalance. The probability of cells being encapsulated into one droplet is low. As a result, the artificial cell type composes a small proportion compared to the whole dataset. As we'll explain later, cluster imbalance will impede the convergence of clustering algorithms. Secondly, ACTs will increase cluster numbers. Given each cell type, the number of artificial cell type could be 2 to the power of h minus 1 minus h. For example, in PBMC, assume we have 8 cell types, then the number of artificial cell type could be up to 247, and the total number of clusters could be up to 255. It's a huge increase of cluster numbers. Thirdly, artificial cell types are not well separated with biological cell types, BCTs. Still in this CD3 versus CD19 scatter plot, we have dodged this ACT cluster in the top right corner. In this condition, we could notice that the B cell type and T cell type are very well separated with each other. After introduction of the ACT cluster, we could notice that there is no clear boundaries between ACT cluster and the nearby clusters. The distance between ACT cluster and nearby clusters are also drastically reduced. Given these three challenges induced by ACT, it's hard for clustering method to perform well in CITE-seq surface marker dataset. As we built our model basically based on Gaussian mixture model, which is usually solved by EM algorithm. Next, I'll explain why these challenges impair the cluster performance, especially in EM algorithm. It has been established that EM algorithm is hard to converge to the global optima if the cluster number is large, or the clusters are close, or cluster sizes are in balance. Well, unfortunately, in CITE-seq surface marker dataset, ACTs induce them all. Formally, EM algorithm is guaranteed to converge if this equation is satisfied. In this equation, there are three key terms. R is the minimum distance between cluster centers. D is the dimensionality of dataset. K is the number of clusters. What does this equation tell us? It tells us if we want EM algorithm to perform better, we could increase the distance between clusters, or we could reduce the dimensionalities, or we could reduce the number of clusters. Previous work also found that EM is hard to converge with imbalanced components. Below I'm showing you a two-component Gaussian mixture. In the balanced case, EM algorithm works well, while in the imbalanced case, we could notice that the estimation by EM is way off. Given CITE-seq dataset which contains lots of clusters, and given ACT clusters is not well separated with other clusters, also given there exists a huge imbalance in CITE-seq dataset, it should not be surprised that GMM does not work well in CITE-seq surface marker dataset. Well, we observe that if we inspect data in lower dimensions, we notice that clusters collapse into each other. It needs to reduce the cluster number and more balanced clusters. Both of them will make it easier for EM to converge. The observation that EM algorithm is easier to converge in lower dimensions leads to our key idea. Instead of taking whole dimensions into clustering at once, why not perform clustering iteratively in lower dimensions? Below I'm showing you the original GMM clustering result in 2 dimensions. Instead of work on 2D dimension, we could also work on single dimensions iteratively. In CD3 dimension, there are clearly 2 well-separated and more balanced subpopulations. After separating these 2 subpopulations, we could also align GMM on CD19 dimension and perform clustering further. By this way, CITE finally separate these 4 clusters, especially this tiny ACT cluster. There are 2 key steps in CITE. The first is components merging, and the second is subspace selection. Although clusters collapse into each other in lower dimensions, they do not always perfectly stack on each other and form perfect Gaussian components. In this case, we have 5 components estimated by BIC criteria, whereas the figure shows some components overlapped largely. It would be not reliable if we separated these components into different clusters. So we adopt Bahatiya distance to measure the overlap, and given a threshold, the overlapped components are iteratively merged into a component complex. In this case, we have two component complexes, which are well separated with each other. In subspaces with more than one component complex, we could conduct reliable subdivision. What if there are multiple subspaces with over one component complex? Heuristically, CITE-sort performs subspace with lowest dimensions. If the dimensionality is the same, CITE-sort will select the one with the best goodness of fit, which is measured by likelihood. Then let's have an overview of CITE-sort. Given a whole population denoted by P0, CITE-sort started by searching subspace and select the subspace to perform bipartition. After the bipartition is conducted, P0 is subdivided into two parts. Then for each part, we check the termination criteria. If the current population cannot be subdivided further or it has too small size, then it's assigned to be a leaf node. Where if the current population could be subdivided further, then we repeat search a subspace and select a bipartition. The process is repeated until all nodes are assigned to be leaf nodes. And all these leaf nodes are just the clusters identified by CITE-sort. In this way, CITE-sort constructs a single-cell sort tree, and each node in the sort tree records the subspace involved in the division. This sort tree could facilitate easy interpretation of the clustering results. Then let's go to the datasets and results parts. We tested CITE-sort in 2 sets of simulation experiments and 8 real datasets. In the simulation experiments, we compare CITE-sort with k-means, GMM, and their variant methods. In the first set of simulation, we fixed the number of cell types as 4 and gradually increased the ACT proportions from 5% to 25%. Adjusted mutual information is used as a metric. As the figure shows, CITE-sort has a superior performance compared to others. In the second set of simulation experiment, we fixed ACT proportion at 5% and gradually increased the number of cell types from 2 to 5. Also, CITE-sort outperforms other clustering methods. As to the results of real datasets, I'm showing you a sort tree built by CITE-sort on PBMC sample. For example, the rule node represents the whole population. It records that CD3 is first selected for subdivision. Then, according to the expression of CD3 in the two subpopulations, we could annotate CD3 positive and CD3 negative in the corresponding edges. Then for this CD3 positive subpopulation, this node record CD8 is further selected for subdivision. Similarly, step by step, we could finally get into the lymph nodes. According to the annotations we have made, this cluster, which is CD3 positive, CD8 positive, and CD19 negative, is just the CITE-seq T cells. Then what is this cluster? It's CD3 positive and CD8 positive and CD19 positive. We cannot match this cluster into any cell type in the list. Actually, it's just the ACT cluster we used as examples previously. In this way, we could annotate all the cell types and the putative ACT clusters in this sort tree. And then we merge all these ACT clusters into a single cluster and highlight it into this T-sim plot. Then we got this figure. It shows ACTs segregated by CITE-sort. If we compare the result with double-light finder, which is a computational method based on single-cell RNA-seq data to infer ACTs, we could see they are consistently with each other. We could also compare CITE-sort to cell Hessian, which is an experimental mechanism to identify some multiplexes. We could see that they are also consistent with each other. This result means CITE-sort can reliably segregate ACT clusters. Let's make a conclusion. CITE-sort is the first artificial cell type-aware clustering method for CITE-seq dataset. The key idea is to iteratively perform clustering in lower dimensions, and we have shown that CITE-sort outperforms canonical clustering methods, and it could reliably segregate ACTs. The clustering process of CITE-sort could be visualized as a sort tree, which facilitates easy cell type annotation. Finally, I'd like to thank everyone who has helped me in this project. Thanks for your listening. I will be happy to take questions if there are any. Thank you.